The cell begins more negative on the inside than outside in its surroundings. Some contributors to this are the fact that K+ ions are more able to diffuse than Na+ ions, as well as the presence of chloride negative ions in negatively charged proteins, as well as the fact that there's Na+, K+ pumps that are consistently working to pump Na+ and K+ ions against their gradients. A neuron is said to be excitable, which means that it will move past its resting potential in charge when enough of a stimulus is received at the dendrites. If it passes negative 55 millivolts, it is said to pass its threshold stimulus, which will cause an action potential. Neurotransmitters being received from other cells are the main reasons why impulses begin. When they begin, neurotransmitters enter the dendrites and enter into the cell body.
As the impulse travels down the axon, it skips the myelinated sheaths and all the action occurs in the nodes of Ranvier. Sodium ions will diffuse through their membranes, switching the charge deposited on the inside, thus creating depolarization. Almost simultaneously, potassium ions will diffuse through their membranes, switching the charges back to its resting potential, which is repolarization. This sequence occurs right after each other. This causes a wave of action potential that travels down the length of the axon to the motor end bulb.
1. As the impulse reaches the motor end bulb, the presence of Na ions causes calcium ion channels to open. The presence of calcium ions makes the synaptic vesicles release their neurotransmitter into the synapse, where they attach to the postsynaptic neurotransmitter receptors on the affected cell.
After being released, some neurotransmitters are decomposed by enzymes. The enzyme acetylcholine esterase decomposes acetylcholine and is present in the synapse and on the postsynaptic membrane of the neurotransmitter neuromuscular junctions, which control skeletal muscle contraction. Other neurotransmitters are transported back into the synaptic knob that released them, or into nearby neurons or neuroglial cells. Decomposition of neurotransmitters prevents continuous stimulation of postsynaptic neurons.